Hello there, and welcome to your notes on the brain. Now, the brain notes are going to be cut up into 3 different lectures, and we're going to first start out with looking at how we know what we know about the brain. So its structure and the way it's physically set up. So this right here is supposed to be funny, but it's looking at the female brain versus the male brain. You can look at that and laugh as you see fit. Okay, so we're going to talk about a couple different things. The techniques look to use inside the brain. So how do we know the different parts of the brain before we go through memorizing the amygdala versus the hippocampus versus the cortex? How do researchers, scientists even know that these different parts exist and where they start and stop and so forth? So we're going to look at 4 different ways that scientists have used to look inside the brain. A couple of them are imaging, so actually looking at pictures, and a couple of them are like looking at electricity and then lesioning. Okay, so we're going to be looking at electrical recordings of the brain, lesioning, simulation, so stimulating parts of the brain and what they do, and then imaging, so actually making pictures of the brain. So let's start with a lesion. So a lesion is like a tissue, like destroying a tissue, or like cutting a part of the brain and then looking at what happens behaviorally when we get rid of that part of the brain. So I'm literally destroying parts of my brain tissue. This could be naturally occurring, or it can be made in a laboratory. So some people, like after strokes or if they have tumors, there's part of their brain tissue that is destroyed, and they really become a case study because I'm looking at what happens when that part of the brain is gone and what behaviors come from it. So these are really case studies right here when we look at strokes, head injuries, brain damage, because I'm not inflicting those on people. Now in laboratories, scientists have been able to purposely lesion parts of animal brains and then looking at what happens because of that. Gardner, he actually worked with humans, but he was looking at different parts of the brains being gone and what type of personalities people have. It was in the 1975s. And then really this is a limited research method because of Mr. Trauber. Ooh, we've magically transported to another place for recording. Okay, so I left off last time talking about lesions in the brain, and we left here with the limitations of them. And really the big limitation about lesioning parts of the brain, although it might show a lot about what a part of the brain does, is that we can really only do it in animals. So it's very experimental to make a lesion in one part of the brain of an animal and see the effects it has on their behavior. And then again, animals, when we're looking at animals versus humans, if we're looking at parts of the brain that have to do with emotion or speech, cognition, animals don't have those same types or parts of the brains that we do. So really those are some limitations. The biggest things we get from brain lesionings with humans are anytime, like again, we talk about that there's a destruction of the brain tissue because there's a tumor and there needs to be something taken out, or old lobotomies that they used to do where they actually remove portions of the brain to treat different disorders. We learn a lot about those case studies, but it's not something that can be replicated on a mass scale. Okay, so now let's talk about how we look at what's inside the brain. We're going to talk about 2 different things. One, looking at waves that occur within the brain with looking at electroencephalograms, so EEGs, and then how we image the parts of the brain. All right, so the first one we're going to talk about is EEGs. This is just recording activity in the brain, so I'm not getting a picture of the brain from this. I'm looking at the brain's activity. Okay, so the EEG, or the electroencephalogram, this is, I think, kind of easy to remember because it has electricity in the word EEG. So no imaging from this, but I basically get an amplified version, or amplified recording of what's going on, like brain waves inside. And they do this by placing these little, what are they called? Electrodes, sorry, on different parts of your head. You may have had one of these before, and then looking at the activity that sweeps across the brain. So this is actually a better picture I skipped over. So this little girl has all these different electrodes, and it's looking at like the different electricity. Remember, in action potentials we talked about that you have those when the axons fire, you get electricity going down those neurons. Okay, so we can actually record different the activity of parts of the brain by putting little electrodes on there to measure it. Okay, now that does not create a picture of the brain. Neither does this. This is a different type of method using electricity. So this is called electrical stimulation, or ESB. This is where they put an electrode into part of the brain, and they stimulate that part of the brain and see what part of the body or the behavior that it affects. All right, so again, most of this is done on animals, as with any type of our experimental brain research, but this can be done with humans. Your brain actually doesn't have any feeling, the brain tissue, so they can actually numb part of the scalp, take it out, and then put an electrode in there. I've seen or heard some of this type of research done with people that have tremors or any type of like seizure disorder, seeing if they stimulate that part of the brain, how that affects the rest of the body. Okay, now let's go on to our imaging. So we're going to talk about 4 different types of imaging where I actually make a picture of what's in my brain. So we're going to look at CT or CAT scans, PET, MRI, and then an fMRI. All right, so those are the 4 ones that actually make an image of my brain, all right, versus the other 2 we're just looking at electrical signals. So the CT scan, or also known as a CAT scan, you might have heard of this. This is called computerized tomography, and it basically is taking a whole bunch of different x-rays of parts of my brain, and then it goes to a computer and it puts it together to make a picture. All right, so you're looking at different slices of the brain, and this is probably the first one that would be done in a medical office if they think there's something wrong, if they're looking for a tumor, because it's the least expensive, it's the least intrusive of the 4 different scans. And this one does show or can show different masses in the brain. So if we're looking for a tumor, it might look something like this. So this is an image from a CAT scan. So this is different parts of the brain, and then this right here would be something that would be abnormal or a growth. So this would be the first thing if I was looking, or if I thought we had a tumor in your head, they might do a CAT scan first to see if there's something vivid that they can see. All right. The second imaging is called a PET scan. This is positron emission tomography. So this one's actually kind of cool. This one is looking at radioactive forms of glucose. So if you remember back to biology, glucose, anytime your body is consuming glucose, it's getting energy. So cellular respiration, scary. But basically your body is breaking that down because it needs energy in that part of the brain. So this type of scan actually traces that radioactive glucose and looking at what parts are active. So you can see these red parts right here and right here. So those are the parts of the brain that are using a lot of glucose at that time, versus the blue parts aren't using a whole lot. This is really neat if I'm looking at, it's kind of like a functional scan, because you can ask people to think of an image or do a math problem, and you're looking at what part of the brain is using the glucose at the time, using energy because it's being worked. Okay, this is done in a bigger machine, looks like this. You might have seen something like this on a movie or Grey's Anatomy or something like that. But you're looking at a PET scan here, so it's actually measuring radioactive forms of glucose. All right, and then the last 2 we're going to talk about an MRI and an fMRI. So these stand for magnetic resonance imaging. MRIs can actually be done on any part of the body because it's looking at soft tissue. So your brain is made of soft tissue. But you can also do an MRI on your heart. You can do it if you've had any injuries, looking at soft tissues in your knee, any part that might be injured. But it's distinguishing among the different types of tissues. So this actually allows us to see the different parts of the brain. Now, a functional MRI, this is where the researcher and the person doing the MRI actually have contact. So again, kind of like a PET scan, where they can say, I want you to think about adding 4 plus 2 plus 7, and looking at the part of the brain that's being used at that time. All right, so here are some pictures of, and it does that by measuring blood flow. All right, so I'm looking at not just where glucose is going, but where my brain is redirecting blood flow to, because it's being used. So fMRIs, blood flow, PET scans is using glucose. MRIs look like this, so I actually can see the soft tissue here. So it creates this computerized model of what the brain actually would look like in the different compartments, right? So you can actually see, this is what, like when you look at a picture of a brain in your book, this is what an fMRI might look like. Okay, bring any questions to class?